Okay, so thank you so much for a kind introduction, Dr. Otsubo. Good afternoon, everybody. My name is Hideki Takahashi. I'm a member of the CFAI of the Tohoku University. First of all, I would like to express heartfelt thanks to all of you present here for the first stage frontier in agriculture immunology. As introduced by the Professor Aso, who is director of the CFAI in this morning, the unique feature of this symposium today is that the researcher who study on the immune system of the livestock and marine organisms and plant come together to meet. And the second session is just focuses on the plant topics of the plant immune system. I'm wondering if many participants in this symposium seems to be studying on the livestock, the fish, immunology of the livestock and fish, and cell fishes. So therefore, before moving to the main lectures, I'm very happy if I could give you an overview of the different and the similarity immune system between the plants and animals for the mutual understanding the differential science background. And then I would like to provide an outline of this session too. When we look close at a major infectious agent which causes outbreak of diseases in the plant and the animals, virus and the fungi and the bacteria have equally potential to infect both organisms. However, about 80% of the plant disease is caused by the infection with the fungi, whereas about 80% of the animal disease is caused by the infection with the bacteria. On the epidemiologic viewpoint, the chemical compound, including antibiotics and pesticides, are equally effective to control the fungal and bacterial pathogens. However, the excessive use of the antibiotics and pesticides causes emergency of drug resistance pathogens. With regard to the virus diseases, in general, it is not easy to control them by the chemical pesticides in both organisms. On the other hand, when we look at the plant and the animals as a host organisms against the pathogen, we notice that there are several differential aspects between the plants and the animals. The plants as you know, the plants are generally rooted in one place and do not move on their own, whereas the most animals have a ability to move freely. From the host cell perspective, there are also the distinct properties between the plant cells and animal cells. As you know, the plant cells are regular in shape because they have a cell wall, whereas animal cells are irregular in shape without cell wall and highly differentiated to perform their function. In the animal immune system, several types of immune cells can move into the body through the vascular tissue and broad vascular and lymphatic system to attack the pathogens. Unlike the animals, the plant do not have phagocytic cells, and in the plant, basically all or most cells at the surface of the leaf and vascular tissue of the stem and the roots have a potential to non-specifically induce the defense response to invasion of any pathogen. Therefore, the mechanism of plant and animal defense system against the pathogen infection have historically been presumed to be separate and distinct. However, discoveries over the past 20 years demonstrated that the mechanisms of plant and animal defense systems show impressive structure and strategic similarity. The immune system protects the plants and animals from the pathogen infection through the layered defenses of increasing specificity. The first line of defense system against the infection is a physical barrier, which prevents the invasion of the pathogen. The waxy cuticle of many plant leaves and the skin of the animal are a good example of these physical barriers. If a pathogen overcomes this physical barrier, the innate immune system provides a mediately but non-specific response. This innate immune system are essentially found in all plants and animals, but now the plant version of the innate immune system is named the plant immune system here. The invertebrates do not generate lymphocytes or the antibody-based humoral response. The leukocytes of the invertebrates, including phagocytes and the natural killer cells. So these cells identify and eliminate the pathogen either by attacking the pathogen through the contact or by engulfing and killing the pathogens. Thus, the innate immune system is a dominant defense system in the most invertebrates. If the pathogen successfully evades the innate immune system, the vertebrates have a second layer of the defense, that is adaptive immune system, through the recognition of specific non-self antigens. The cells of the adaptive immune system are lymphocytes, and the B cell and the T cell are the major type of the lymphocytes, and are derived from the bone marrow and the thymus. The B cells are involved in the humoral immune response, where the T cells are involved in the cell-mediated immune response. On the other hand, the plant immune system, plant responds to pathogens infection using a two-stepped immune system. The first is pathogen-associated molecular pattern-triggered immunity, that is PTI. And the second is effector-triggered immunity, that is ETI. In the PTI here, the plant recognizes the molecule common to many classes of microorganisms, including non-pathogens, and activates the defense response. And to overcome this PTI, the pathogen secretes the effectors to the host cytoplasms and prevent several steps in the signal transduction pathways, confining the defense reactions here. However, the special resistance variety of the host plant can detect this effector as a molecular signature by immune receptor, and again activated the downstream signal transduction pathway and produce antimicrobial compounds. So this second defense response is named the effector-triggered immunity, the ETI. Here, I would like to emphasize that the innate immune system is an evolutionary order defense strategy and seems to be diversified as the dominant immune system in the plants, insect, and primary multicellular organisms like shellfishes. The fact that the plant immune system and the animal innate immune system share a common origin. The fact seems to be supported by impressive structural similarity of the pattern recognition receptors that detects the molecular signature of the pathogens. For example, the leucine lipid-containing receptor kinase and the NBLRR class intercellular immune receptor that mediate awareness of non-self and are integral to the host defense in plant and animals equally. Furthermore, signal transduction pathways at the downstream of pattern recognition receptor have strategic similarity in the plant and the animals. Of course, the individual component of the signal transduction pathway is not completely conserved between the two organisms. But for example, the eukaryotic the MAP kinase cascade seems to play a key role defense signaling of plant and animals. Another common defense system against the pathogen infection between the plant and animal seems to be a virus-induced RNA silencing. What you can see here is just overview of the RNA silencing pathway. The disoprotein family here is a key component of this pathway and diversified during the evolution of the plants and the animals. So this fact indicated that the plants and the animals have evolved a similar defense strategy against the virus infection. And by the way, as you know, adaptive immune system create immunological memory after an initial response to the specific pathogen. This process of the adaptive immunity is a basis of the vaccination. Interestingly, on the plant-pathogen interaction, we can see the similar phenomenon, which have been named induced systemic resistance or priming response. Another interesting aspect seems to be non-pathogenic microorganisms mediated activation of the immune system in the plant and animals. The metagenomic analysis of gut bacterium seems to be a recent popular topic in the animal immunology. As well, the analysis of microbiome in the rhizosphere of the plant get considerable attention now. In this second session, these four speakers could give an excellent talk about plant immune system and infection strategy of fungal pathogens. I'm wondering if the Professor Corne Petersen of Utrecht University could give a talk about induced systemic response, induced systemic resistance, excuse me, and root microbiome. And Professor Kang of the Texas University could give a talk focusing just the role of the transposable element on the epigenetics in the plant defense signalings. And then Professor Kubo of Kyoto Prefecture University could talk about infection strategy of plant pathogenic fungi and plant immunity. And the last talk, Professor Komerik of the Wageningen University could mention the antiviral defense system against the Gemini viruses here. So finally, I would like to greatly appreciate all the speakers who could accept to give a talk with good grounds. And I'm very happy if all the participants could have an active discussion in this session. Okay, thank you so much for your kind attention. And we would like to move the first talk of the Professor Corne Petersen.